Hello everyone and welcome to my channel Science Query, and in this video I am going to talk about glucokinase, the role of glucokinase in glycolysis and what is the difference between glucokinase and hexokinase. Glucokinase is the first enzyme used in the step of glycolysis. It helps in phosphorylation of glucose to glucose 6-phosphate. Glucokinase is expressed in cells of liver and pancreas of humans and most other vertebrates. In the cytosol of the cell, glucose enters through GLUT2 transport. This glucose then further metabolized by either using pentose phosphate pathway or by glycolysis to produce energy. So glucose enters through GLUT2 channels and undergoes further metabolism. The first step is the irreversible reaction step, where the enzyme glucokinase and ATP reacts with glucose, and it marks the beginning of energy investment phase, producing ADP and glucose 6-phosphate. This glucose 6-phosphate then either goes to pentose phosphate pathway or continues to glycolysis. Now let us understand about the enzyme glucokinase and how it works. Glucokinase is also known as hexokinase 4. It is the first enzyme that functions as a primary glucose sensor in the mammals and plays a crucial role in maintaining blood glucose homeostasis. It catalyzes the glucose in the cytosol by adding phosphate group in the 6th carbon of the glucose. The enzyme glucokinase combines with substrate, that is glucose and ATP, and the phosphorus group from the ATP transferred to the 6th carbon of the glucose, converting it into glucose 6-phosphate and ADP. This reaction also requires the magnesium ion, which stabilizes the negative charge on the phosphate group of the ADP. The presence of the phosphate group makes the glucose molecule less stable and more reactive or active it for the further step of the metabolic pathway. In simple terms, we can say that the cells spend ATP as a currency with the help of glucokinase enzyme to trap the stable glucose and ensures that it cannot escape and is ready for the further energy releasing steps like pentose phosphate pathway and glycolysis pathway. Next, let us see what is the difference between glucokinase and hexokinase enzymes. The hexokinase enzyme, which initiates the glycolysis, is having 4 different types of isomers known as hexokinase 1 or HK1, hexokinase 2 or HK2, hexokinase 3 or HK3, and hexokinase 4 or HK4. The similarities of all these 4 isomers of hexokinase are all of them helps in catalyzing the irreversible first step of glycolysis. All of them involves in the phosphorylation of the carbon in the 6th position of the glucose molecule. The hexokinase 4 or HK4 is also known as glucokinase enzyme. The others are called hexokinase enzyme in general. The basic differences between hexokinase enzyme and glucokinase enzymes are as follows. HK1, HK2 and HK3 are the examples of hexokinase enzyme, and HK4 is known as the glucokinase enzyme. Hexokinase enzymes are mainly found in muscle, brain and red blood cells, whereas glucokinase enzymes are present in liver and pancreatic beta cells. The Km for glucose is low for the hexokinase enzyme, and the Km for glucose is high for the glucokinase enzyme. Now let us try to understand what is Km for any particular enzyme. Km, or the Michaelis constant, is the measure of the affinity of an enzyme for its substrate, with high Km value indicating a lower affinity. Let us see the graph for a particular enzyme for substrate concentration at one end and the rate of reaction or the velocity of reaction at the other end. At zero substrate concentration, there will be no reaction. With increase in substrate concentration, there is an increase in rate of reaction. The initial rate of reaction is always steep, that is, it always high and it always grow fast. But after some point, if we keep increasing the substrate concentration, the steepness becomes slightly less. And at some point, it remains same even if we increase the substrate concentration. This maximum rate of reaction is known as the Vmax. Vmax varies with different enzymes and temperature. For any reaction, half of the rate of reaction is called half Vmax. And at half Vmax, there will be a specific substrate concentration. When you draw a straight line down, this is known as the Km of that enzyme of Michaelis constant. Let us compare the Km value for enzyme A and enzyme B. Here, for example, suppose the Km for enzyme A is 8. That is, if there are 4 enzymes present in the reaction, out of which 4 enzymes, half of the enzyme sites are occupied by the substrate when its concentration reaches 8. That is, 2 molecules occupies the enzyme and the remaining 6 are free. Now suppose the Km for the enzyme B is 10. That is, if again there are 4 enzymes present, to reach the half Vmax, 10 substrate molecules are required. To reach the half Vmax, out of 10, 2 of the substrate molecule engage with the enzyme and other 8 remain free in the solution. Hence from this graph we can say that the higher the Km, the lower is the affinity of the substrate towards the enzyme, and lower the Km, the higher is the affinity of the substrate towards the enzyme. Affinity for glucose is high in case of hexokinase enzyme and the affinity for glucose is low in case of glucokinase enzyme. The Vmax of hexokinase enzyme is low, that is, it has low capacity, whereas the Vmax for glucokinase enzyme is high, that is, it has high capacity. Hexokinase enzymes are known as housekeeping enzymes. They ensure that the glucose uptake for energy even when the blood sugar level is low. The glucokinase enzymes are known as the glucose sensor. They act only when the blood sugar is high after a meal to store or dispose of excess glucose. The inhibition in hexokinase enzymes are allosteric inhibition, whereas the inhibition in glucokinase enzymes are mostly regulated by insulin hormone. Well, that is all about the glucokinase enzyme. If you like my video, then please subscribe my channel Science Query and please like, share and comment. Thank you.